Hey everybody, last Friday Vivian and I were talking. I think we were having some good ideas, but as much as I wanted to, I couldn't really explain how everything I was talking about fit together and what it would look like in real life. So this is kind of an attempt to show you a big picture idea and a flushed out part of a small piece of that big picture. So this is a Prezi, and there's a link to it in the email that I sent you. Now, around the outside is kind of the sequence of events that we've talked about, and in the inside is a central theme. Around the outside of the center ring, this ring, are ideas that might connect to research, Tripp's class, Francine's class, and perhaps a neuroscience class. Okay, so let me start by saying that this is only my idea and that I'm really looking for feedback on this. I don't quite expect it to work, and I expect it definitely to change. So we begin this with Michaela's project, her kind of open inquiry one. We move on to Adam Smith, which is a project that we would use to teach intermolecular forces and basic biochemistry. We then move on to macromolecules, molecular machines, central dogma, epigenetics, the environment, and how those all affect gene expression. And then we kind of move on to these big picture ideas. Um, this is really only flushed out to about this point right here with the Adam Smith project. The interesting thing I wanted to show you here is that from macromolecules and classification, we can go to use uh, the concepts that we've taught here to learn about HIV vaccines, to learn primer design, which would then later aid us in PCR and create a tie-in to TRIPS class. We can also use those concepts to learn about nutrition labels. And right here, a project I want to call the Munchies, which I think would be perfect for Francine, talking about CB1, which is a brain receptor, and the ability of scientists to create drugs that bind to it and how those have been affecting nutrition, health, and specifically the idea of hunger. Great, so let's go into and look at what the Adam Smith project might look like. So Adam Smith is a computer program made by a company called BitTwixt, and it's based on the idea of molecular design, or dynamics, which is a fast-growing field in which computers model chemical reactions, basically so that chemists don't need to do them. It's being used a lot in drug design, and it's great for 3D modeling and produces really accurate images in the kids' heads of molecular ideas. So this is Adam Smith. It's a program that we can buy a site license for so that every kid can have on their own computer. Um, on the right here is our box builder. This is going to allow us to control this box, which is a simulation. So let me give you an example of some things we can do. We can put a bunch of molecules into this box, and we can see how they interact. We can raise the temperature, alter the processing speed. We can change the size of the box. And then we can also do things like look at London dispersion forces, hydrogen bonds, and such things. So it really allows kids to play around, and they can alter a bunch of different things. And at the same time, what they're starting to learn is that molecules are not these static things that we draw on the board with lines, O's, and H's, that they're these moving, wiggling things that interact with each other. I'm going to do a quick water demo just to aid in our talks later. So we're going to go ahead and start this up. Going to go ahead and speed it up. And then I'm going to go ahead and drop the temperature. So what you should see pretty quickly is that the water will start to stick together. And then we're going to slow this down again, put on some hydrogen bonds, and then take a closer look at what's going on. So this is, some things that I really like about this program. But what we can see here is that you have a whole bunch of interactions. And what a kid might say is that oh yeah, these waters are sticking together, and upon closer examination, they might be able to say the hydrogens, the white hydrogens, look like they're sticking to the red oxygens. So then the question becomes, how do we use that in our class? Well, so we have the kids play with Adam Smith. We have them make whatever observations they want to make. We have them describe whatever patterns they want to describe. And we take them basically through the process of experimental design. They look at those patterns, they attempt to describe those patterns scientifically, they see if those patterns hold. For the case of water, we look and see if maybe hydrogens stick to everything. We go ahead and put the water in with other molecules, and then we find it sticks to some things and doesn't stick to others. The kids will then go and come up with a theory for why this behavior is happening. Why do oxygens and hydrogens seem to stick together? Well, we know because the partial charges of hydrogens are positive and partial charges of oxygens are negative, and those two things will come together because of electronegativities and thermodynamics, but the kids will actually generate all those ideas on their own. So they will teach themselves through this the universal gas laws, intermolecular forces, and thermodynamics. The reason I really like this is because it gets the ideas of the ball and stick O&H chemical reactions out of their heads, but it gives them very good 3D ideas of what is happening. It also gets out of their heads the idea that chemistry and biology aren't linked, and if we extrapolate this a little bit further, it will get them to show them that chemistry does matter to them, and that chemistry does matter to them. So clearly what we're teaching here is some experimental design. We're teaching them the basics of the biology class that we want to teach them. And if we take it a little bit further, so we go to macromolecules and we use these ideas and this program to teach intermolecular forces, binding, for example, then what we can do is we can have the students start to design primers as a test to see whether they understand the concepts of thermodynamics and intermolecular forces. We can have them talk about research antagonists with Francine, and we can outlink this to crazy ideas like DNA origami, which is kind of hot in the world of science right now. So these two smiley faces were made by scientists at Caltech. They were made out of DNA and were coded for and are kind of the basis behind a lot of nanobot type of ideas. Just thought I would show that to you. So then the question becomes, how do we evaluate these things? Well, I thought it would be interesting to have the students kind of write their own textbook as we go. So after they've done this Adam Smith project, they've generated the ideas of thermodynamics, they teach each other the lessons they've learned based on the patterns that they themselves came up with. And each student generates their own textbook to make sure that they're understanding what we want them to understand. If they do indeed understand all the ideas we've talked about, then they should also be able to design primers and predict folding patterns that we can evaluate. And that brings us to our template. So this is a template really specifically designed only for the macromolecules unit, the biochemistry unit and macromolecules unit, and is the basis of the much larger template. Again, this is just what I'm putting forth as my idea. Hopefully we will change it and make it better based on everybody's ideas. But the core concepts in the content area are the kind of chemical nature of biology. First of all, that charge and determines intermolecular kind of interactions. So positive and negative things stick together. And then at a higher level, that electronegativity and intermolecular forces help to predict how much larger molecules will happen. And the idea that much larger molecules like DNA and proteins fold based on these principles. We have the idea kind of coming from macromolecules that macromolecules are based off of these monomer units that these macromolecules classes kind of macromolecules behave in groups of ways. So, for example, lipids behave in a way, amino acids or proteins behave in a certain way, and DNA behaves in a certain way, or nucleics behave in a certain way. And finally, at a really high level, that emergent properties come out. So you have a molecule based off of these little monomers. Now, these large molecules act in a certain way, but really interestingly is that these macromolecules become or have much greater properties than their small constituents. The experimental design aspect, I think, is fairly straightforward. The ability to recognize patterns, basically the ability to scientifically describe those patterns, and then at an exemplary level, the ability to find out why those patterns exist. So I see that water is sticking to each other. Oh, that water sticking to each other really is an attraction between a oxygen and a hydrogen. Oh, my electronegativity will make my oxygen partially negative, my electronegativity will make my hydrogen partially positive, and that's why I'm going to get those relationships. Once I've got that kind of theory going, I start to design an experiment in which I identify the one thing I'm going to change. Well, if my partially negative hydrogen is attracting to my partially positive oxygen, and that is what is driving the interactions, then a partially negative oxygen will not be attracted to a neutral object, which helps me to generate my theory and my experiment, and then a recognition of positive and negative controls, basically the ability to generate negative controls. There's more to it than that, but hopefully this will give you a good idea of what I was talking about and how I plan to evaluate everything and how it all looks as a class.